So poliovirus is actually a problem that's an endemic in areas where there are unsanitary conditions, and also poliovirus infects the very young or the very old or immune compromised. And in many of the countries, those people don't get proper meals, they're malnourished, and are very susceptible to poliovirus. It's endemic in Nigeria, it's endemic in Afghanistan, Pakistan, and northern India. But there is spillover in all the countries that surround those. There's cases of reemergence of polio in these areas because there's lack of vaccination. And you have importation of cases where people travel who have polio. I think people need to become a little more aware that this is a reemerging virus, reemerging disease. And this is primarily because we dropped our guard and we haven't fully done our due diligence in vaccination. The reason why you can eradicate polio is the virus is not very stable in the environment. Humans are the only host for the virus. We know we can vaccinate and produce long-term protection, and we know we can make the vaccine. So essentially there's two forms of vaccines. There's oral polio, and then there's, which is the Sabin vaccine, and there's the inactivated polio, which is the Salk vaccine. Both vaccines are very effective. However, the oral vaccine requires multiple doses. In this country, it requires at least 2 to 3 boosts, as an example. In countries where there's endemic polio, it can be as high as 8 to 12 boosts. That means you have to go back and target those people you already immunized to get them the number of boosts. In a country which there's hundreds of millions of people, such as in northern India, this becomes a logistic nightmare. So the goal here is to create a vaccine that has high potency and can be administered once, maybe twice, to achieve the same level of efficacy. And this is where the inactivated vaccine comes in. Well, the primary problem in all vaccines is cost. And that's the reason the Gates Foundation is interested in this, because the current cost for making vaccines for a most efficacious vaccine is an inactivated polio vaccine. And that's 15 times the cost of an oral polio vaccine. So as soon as you change the manufacturing process, that adds cost. And we don't want to do that. We're trying to lower cost and increase distribution. So one of the things we do here is we're one of the centers of excellence for influenza research and surveillance. And part of our pilot project in that area was to utilize what's called an SI genome library. And this is a library of RNAi molecules that can target every single gene in the human genome. And it allows us to ask very specific questions like, what role does this host gene have in virus replication or in resistance from virus replication? So we know viruses co-opt host cells to replicate. And similarly, host cells have features or gene methods to prevent viruses from replicating as well. So if we go after those genes that resist virus replication, we can therefore make vaccines. And we tested this possibility with flu and found that we can actually make enhanced vaccine cell lines for flu using mammalian cells and increase the flu production by about 10,000-fold, 1.5 logs, which is pretty amazing. So that means we can use 1 egg instead of 10,000 eggs to achieve the same level of vaccination. Our group, in fact, was the first to use RNAi to make an antiviral that was against RSV. That's now in phase 3 clinical trials. So we're taking that kind of technology now and working towards vaccine development. The goal here is to go after those genes that resist viruses to allow for increased virus replication to make more vaccine and reduce cost. So the Gates Foundation is all milestone driven, and essentially what we have is a year to discover the genes and make the first validation that those genes are important. After that, we move towards the validation step of doing this on a large scale for vaccine production, where we would send the cell line to India and we'd be characterizing, for example, the antigen produced locally, such as in a vaccine testing unit, which are at Emory and Vanderbilt and other places, and they would be testing it as well over in India. So in a period of 2 years, we will have the vaccine that's made in a vaccine enhanced cell line ready to go. So the grand challenges of Gates is all about sustainability. They don't put money into a project that'll be a one-off kind of a thing. Sustainability means we're going to develop a foundational technique that's going to change the paradigm of how vaccines are made. We're going to be able to take and make enhanced vaccine cell lines, not just for polio, but for measles, for rotavirus, for flu, and so forth. So any vaccine preventable disease will go through this process. And we're actually already funded to do the polio, but the next in line is the measles, and we then have rotavirus right behind that. So from the CDC's perspective, they're full on board. From industry's perspective, they're full on board because they don't have to change the method by which they make vaccines. We're just giving them a better product. And then from our perspective, we're not only learning a lot about how to make better vaccines, but the other side of doing all this work is we find out that there's these genes in the host cells that are absolutely required for virus replication. So this leads us now to new disease intervention opportunities where we can target those genes with antivirals. So it's a very powerful approach, and that's the sustainability part of the Gates funding.